Every time we have a headache, sore throat, or cramps, our mind tells us to turn to any over-the-counter medicine we can get. How many pills should you take? As many as you want? One for every hour that the symptom has lasted? Of course not. In the 1900s, the norm was to take a medicine in higher and higher quantities until toxicity set in. This, of course, worsened symptoms and persisted illnesses for many patients. When doctors finally drew connections to these dosage requirements, scientists were able to determine what measures could ensure that patients safely and effectively took the medicines they needed. Let's take a look into a time in history when doctors lacked information on a drug we're all familiar with, aspirin. In 1917, Bayer, aspirin's creator, lost their patent in the US, creating an easier and cheaper access to aspirin for drug manufacturers. In 1918, amongst the terrifying influenza pandemic, many doctors looked to aspirin as a treatment to suppress symptoms of the flu. Doctors and manufacturers still had yet to establish and set safe dosages for aspirin. Thus, aspirin's bottles did not contain any warning for possible toxicity and held minimal instruction on proper usage. Doctors and manufacturers made official recommendations for those affected by the flu to take a dose of 1,000 milligrams of aspirin every 3 hours for reference. It is about 3 times the amount recommended today to be taken in a larger time span than 3 hours, 4 to 6 hours. Throughout the year of 1918, fatalities rose in many young adults. Scientists in the 1940s discovered that in an adult who died from aspirin poisoning, there was a 46% incidence of pulmonary edema. Further studies found that aspirin increased lung fluid and protein levels, which prevented the body from clearing mucus. This served as an explanation for why the causes of these deaths appeared to be caused by the flu. Pharmacokinetic data available today shows that the regimens recommended for the Spanish influenza outbreak in 1918 predisposed people to severe pulmonary toxicity, which could have easily been avoided had there been proper dosage suggestions and labeling on the drug bottles. Over the next several years, public health crises began to emerge as a result of a lack of information surrounding drug dosaging. In 1962, the FDA instituted the rule that new drugs were now mandated to undergo Phase 2 trials in drug development in order to have substantial evidence of drug safety and efficacy. During Phase 2 of these trials, small toxicity trials are performed on healthy volunteers. Phase 2 represents a time where actual patients test the drug and previously unknown side effects from toxicity are discovered. They are used to find the proper dosage for the individuals, which is later used for larger groups of people during Phase 3. For Phase 3, researchers give the drugs to larger groups of patients in order to confirm the efficacy of the drug and see if any new side effects occur. During Phases 2 and 3, between 30 and 58% of drugs fail and are either proven ineffective or unsafe. The introduction of drugs phases by the FDA has allowed for us to know what is safe and effective in the drugs we take. When we visit the doctors, the information from these trials is used in order to calculate a safe and effective dose, as well as warn us about any side effects to watch out for. The development of drug labeling and dosage helped advance over-the-counter and prescription medicine only after many health crises from many medicines in the 1900s. The mistakes we have made in the past have paved the way for an efficient dosage system that works toward benefiting the patient and their individual needs.